Well, hello and welcome to this first webinar in the ISHR Asia Pacific Cardiovascular webinar series. It gives me great pleasure to welcome our two speakers, Professor Louise Burrell from the University of Melbourne and Austin Health, and Dr. Adam Hill from the Victor Chang Cardiac Research Institute. My name is Kate Weeks and I'm a researcher at the Baker Heart and Diabetes Institute in Melbourne, and my co-host, who you can also see on the screen, is Dr. Jim Bell from La Trobe University. So the topic of today's webinar is cardiovascular research in the time of COVID. And we have invited Louise and Adam to tell us about their research, but also to comment on how the cardiovascular research landscape is changing in light of COVID. Before we begin, because this is the first webinar in the series, I would just like to draw your attention to the two links that you should be able to see on your screen. So the first is a link to the program of upcoming talks, and the second is a link to the ISHR YouTube channel, where you should be able to view recordings of these webinars from the Asia Pacific series, but you can also view all of the webinars from the European and American section webinar series, which was launched last month. So I encourage you to check out both of those links. I'd also like to take this opportunity to acknowledge ISHR International for their support of this initiative. I'd also like to thank the Baker Institute for hosting this Zoom webinar, and also the Australian Cardiovascular Alliance, who have been very enthusiastic and supportive of us setting up this webinar series, and who have helped us to reach out to members of the Australian cardiovascular research community to get involved in the series. So I'd now like to introduce Dr. Adam Hill, who is head of the Computational Cardiology Research Group at the Victor Chang Cardiac Research Institute, and he is Deputy Director of the Victor Chang Cardiovascular Innovation Centre. So Adam received his PhD from Imperial College London before joining the Victor Chang in 2005 as part of the Mark Cowley-Lidwell Research Program in Cardiac Electrophysiology, and he founded his lab there in 2011. So a major focus of Adam's current research is using in silico modelling and in vitro pharmacological assays to better understand drug-induced arrhythmias, and his work developing improved preclinical screens for pro-arrhythmic risk has led to invitations to sit on committees advising the US Food and Drug Administration on the development and implementation of a new approach to preclinical safety screening centred around computational modelling of arrhythmic risk. So thank you for joining us today, Adam, and please start whenever you're ready.
So thanks for that introduction, Kate, and thank you to the ISHR Australasian section for the invite to be part of the launch of this new Asia Pac webinar series. As Kate just said, one of the major focuses for my lab over the last 5 years has been understanding the how and why of why certain drugs cause heart rhythm disturbances and developing better preclinical screens for proarrhythmia. And it turns out this is an area that's taken on an increased degree of significance with recent efforts to repurpose drugs to treat COVID-19. And we realize we have quite unique technical capacity as well as the range of skills to respond pretty quickly on assessing the cardiac safety profile of these drugs. And that's what I'm going to talk to you about today. And I'd emphasize that everything I'm going to show you is really hot off the press in the last 6 weeks or thereabouts. So it's very much a work in progress, perhaps a little rough around the edges, but I thought it would make an interesting and topical focus for this webinar. So as a background, in the overwhelming majority of cases, acquired or drug-induced long QT syndrome occurs when the drug blocks hERG, which carries the IKR current, one of the major contributors to repolarization in the heart. And as a result of block of IKR, we get prolongation of the action potential, and sometimes the emergence of these potentially proarrhythmic events, such as early after depolarization, shown here in red, where there's ectopic depolarization of the membrane. And on the surface ECG, delayed repolarization manifests as prolongation as the QT interval, and under just the right circumstances of timing and electrical coupling, EADs such as this can initiate a potentially fatal arrhythmia, torsade de pointes, which is characteristic of QT prolongation disorders. What I want to highlight here is that this is not some boutique side effect of 1 or 2 drugs, but some estimates put it at 70% of all compounds in development, which are thought to block hERG at low micromolar concentrations. And this is one of the main reasons that lead compounds have their development terminated in the preclinical phase. So this problem really came into focus in the 1990s when several antipsychotics of these side effects. And as a result of this, a range of preclinical guidelines were implemented that mandated that all compounds in development have to be tested for hERG block, as well as action potential or QT prolongation. Now, there are some problems with this approach to screening, and more it's being recognized as a fairly blunt tool, which I'll touch on a little bit later. But nevertheless, they've proved very successful in that no new pro-arrhythmic drugs have unexpectedly come to market since they were introduced in 2005. So what does this have to do with COVID? Well, as with many things today, this is an area that's taken on a high profile since Donald Trump tweeted about it and spooked the effectiveness of hydroxychloroquine, an anti-malarial drug in combination with azithromycin for treatment of COVID-19. So here's his tweet. Hydroxychloroquine and azithromycin taken together have a chance to be one of the biggest game changers in the history of medicine. And you'll probably notice that he's doubled down on this in the last couple of days and told everyone he's been self-medicating with these drugs. But unfortunately, it isn't based on particularly strong evidence so far. And the problem in terms of today's talk is shown in this table here, where both hydroxychloroquine, the related drug chloroquine, and azithromycin are all listed in the Credible Meds database as known to cause arrhythmias. And there are hundreds of instances of arrhythmia, as well as cardiac arrest in FAERS, which is the FDA's adverse events reporting system. So both of these drugs individually can cause arrhythmias, and of course together that effect could be amplified. So to go back to the science and ask what the evidence was that these drugs might be useful in treating COVID-19. This data this in vitro data was published a couple of months ago now. They're infecting vero cells with the virus and adding either chloroquine or hydroxychloroquine for various periods of time and measuring virus in the supernatant by PCR. And what you can see is that both drugs, at least in this in vitro setting, effectively inhibit the virus. But one thing I want to draw your attention to is the effective concentration. So up to tens of micromolar here. And this is in comparison to what might be used for a weekly prophylactic dose for malaria, which might hit a Cmax 50 or 100 times less than this dose that the in vitro tests suggest might be needed for COVID. This paper then came out reporting the use of hydroxychloroquine in treating patients with COVID-19. It was quite controversial. This initial report only had 20 subjects, although they've since extended that to a larger cohort. And there was some criticism about inclusion criteria and other factors, but nevertheless, this was the result that caught the headlines. So the black line shows PCR positive patients in the control group. Blue shows PCR positive in the group treated with hydroxychloroquine. And green shows patients treated with hydroxychloroquine and azithromycin. And they're claiming that there were no patients with detectable virus on day 5. Now, subsequent studies have shown certainly mixed results, and in particular in sicker cohorts, there's no efficacy has been demonstrated. But off the back of this, there's now more than 200 trials registered for hydroxychloroquine, many centers reporting using it off-label. So this has prompted this growing concern about the safety profile of these drugs, particularly at the doses required for COVID-19. So back to that issue of cardiac safety. For the currently approved indications, these drugs have an incidence of cardiac effects of about 1 or 2%. So that's not necessarily arrhythmia, but it could be just significant QT prolongation. So 10,000 affected individuals per million. The issue we potentially face with COVID is that if these trials were successful and these drugs were even given as prophylactics, they could be given to a significant percentage of the world's population. So this risk group could swell very quickly. And this risk profile is potentially even more complicated in the context of COVID. So this recent report in JAMA Cardiology showed significant QT prolongation in 30% of patients, with 20% reporting QT intervals greater than 500 milliseconds, which is commonly considered a red line for risk. So a much higher instance than the non-COVID use. And the authors also noted there was particularly serious side effects in those patients with renal dysfunction. So with that in mind, we set out to assess the cardiac safety profile of these drugs at doses relevant to COVID and to examine how this was modified by COVID-related risk factors. So here's the specific approach. First, to measure the potency of block of hERG channels by the drugs, both as individual compounds and in combinations. Determine how COVID-associated risk pH changes that might occur with acidosis and temperature associated with fever might modify the degree of block by these drugs. And finally, using silico simulations to model the impact of COVID-relevant doses of the drugs on populations of genetically diverse individuals with and without these environmental modifiers. To achieve the first of those aims and measure the potency against hERG is something we usually do with patch clamp electrophysiology. So we can take a CHO cell expressing the hERG protein, attach a microelectrode and record hERG current, and we can measure the current amplitude as shown here. We can then put on the drug and measure the reduction in peak as an indication of the degree of block, and from that we can create dose response curves for block. And here's a list of the conditions that we were looking to test. So physiological saline, low and high potassium, low and high magnesium, a couple of fairly severe degrees of acidosis. And for each of these conditions, dose responses are to be measured for all three drugs alone and in combination at both room temperature, physiological, and febrile temperatures. So this equates to acquiring data from about 12,000 individual cells. So practically, it's not going to happen with manual patch clamp unless you literally have years to do this. And clearly in the case where we want rapid evaluation for a repurposed drug, we don't have that time. Well, luckily in the last few years, there's been development of some very high throughput automated patch clamp systems. The one we have at Victor Chang Innovation Center is the SynchroPatch PE384, where the 384 refers to the capacity to record from 384 cells in parallel. And critically for our experiments, the platform also integrates liquid handling, so we can screen all those conditions and drug combinations in an automated way, as well as change the temperature for our experiments. This is what the data looks like on screen. So each of these green squares is an individual experiment, and essentially what this means is that we can gather our dose response data in sufficiently high throughput to meet our requirements. And I'm not going to go into all the data today, but just show you a few key observations. So first, here's the data describing potency of each of the drugs against hERG at physiological temperature. And you can see that all three compounds block hERG with micromolar IC50s, with the half maximal dose for azithromycin being about 100-fold higher than the other two drugs. And what I've superimposed here is the estimated range of therapeutic Cmax for each of the drugs based on the doses being proposed in various clinical trials. And you'll also recall that that effective in vitro dose, tens of micromolar, was right at the top of this range here. So it's very clear that at the doses being proposed to treat COVID, there's likely substantial hERG block and with it the potential for pro-rhythmic risk. So how are those potencies modified by some of the metabolic change we see in COVID-19 patients? So firstly, here's the data for kalemic variation. High K, so 7 millimolar potassium didn't affect potency for either drug, whereas hypokalemia, 3 millimolar, millimolar, not nanomolar potassium, increased the potency for hydroxychloroquine. And the data for chloroquine here is a little ragged. There's something not quite right with the steepness of this hill slope. So we'll delay interpretation of that while we tidy it up. But in terms of why we might see this kind of increase in potency, one explanation is that it may reflect the preference of these drugs to bind to the inactivated state of the channel, as many drugs do. And since lowering the potassium shifts the channel equilibrium towards the inactivated state, this means we'd see more block for a given concentration. Acidosis, on the other hand, has the opposite effect. For both drugs, lowering pH very strongly reduces potency against hERG. And we consider whether this effect might be down to the protonation state of the drug, but neither really has a functional group with a pKa that would really change its charge over this pH range. So we're thinking it being more of an effect on the channel and a modification of the binding site. And I should point out that while these are relatively small absolute changes in pH, in terms of pathophysiology, they're very significant. So pH 7 would be very severe respiratory acidosis, while 6.5 would only really be seen locally in the myocardium in the context of a severely occluded coronary, for example. Finally, temperature, which we're looking at in the context of fever. I realize this assay can't capture the complexity of COVID-associated fever like the cytokine storm, so we're solely looking at a purely temperature effect on the drug channel interaction. In our previous publications, we've shown very different temperature effects for the dependency of hERG. This whole chloroquine, whereas increasing febrile temperature shifts the potency to a lower potency for chloroquine. And while I don't have the complete data set for azithromycin here, what we do have so far suggests that this is different again, with high temperatures actually increasing potency. So for these temperature effects, they are complex and potentially drug specific. So just to wrap up that first part of the talk, I've shown you significant block by chloroquine and hydroxychloroquine at doses being proposed in clinical trials. And this suggests a significant pro-arrhythmic risk profile for these compounds in the context of these doses. I've shown you there's complex interplay of COVID risk factors with hERG potency, so pH or low pH and temperature both reduce potency, while electrolyte imbalances can increase potency, and demonstrate that rapid screening on a good time scale of repurposed compounds in the context of all these environmental modifiers can be achieved using synchrotase. So we're probably looking around somewhere in the region of a week or two for an individual drug based on what we've done so far. Now, as I mentioned earlier, hERG block alone is increasingly being considered as an imperfect surrogate for risk. And while I think we've ameliorated some of that inaccuracy by broadening our assay to include the environmental modifiers, we still want to think about how we can best interpret hERG block in the context of cardiac electrophysiology. And this has been a pretty hot question in safety pharmacology circles for the past 5 years, and it's prompted the FDA to propose CIPA, the Comprehensive In Vitro Pro-Arrhythmic Assay, which is a major collaboration between industry, regulatory and advisory bodies, and academia, including us. And it aims to come up with more accurate predictors of pro-arrhythmic risk than hERG block alone. And to achieve this, it's split into two complementary components, a combination of computer simulations and stem cell-derived cardiomyocytes as a validation platform. And specifically on this side of the coin, this involves integrating in vitro measures of ion channel block into computer models of ventricular cell electrophysiology that can then make predictions about proarrhythmia. So I think this gives us a framework to interpreting hERG potency measured in the screens that I've just shown you in a more physiologically relevant context. When we talk about in silico drug screening, what do we really mean? Well, here's a schematic of the myocyte with the membrane in blue, all those channels and pumps that move current across the membrane shown in gray. And in the computer model, each of them is represented by a series of equations which describe the channel function in terms of the transitions between closed, open, and inactivated states that occur in response to voltage. And when we've got a fully coded model, we plug that into the computer and we can output an action potential such as the one shown here. Now, in terms of predicting the effect of a drug, we can measure a drug's potency against specific cardiac ion channels in a screen, as I just showed you in the first part of the talk. And those measures of block can be incorporated into the model simply by scaling the relevant ion channel conductances. And then this can be interpreted by the model to predict an effect on the action potential, typically measured as a change in the duration of repolarization at 90% return to baseline, or the APD90. And we can actually take that a leap further, and rather than just predicting the effect of the drug in a single action potential, so representative of an individual subject, we can extend the model to predict population drug responses. And to do that in silico is relatively simple. So here's that baseline model I just showed you in outputs and action potential as shown here. And we can add variation, either up or down regulation, to the expression of all the rhythmonome genes using scalars taken from a randomized distribution. And when we do that hundreds or thousands of times, we end up with this population of action potentials, with each one representing an individual with a different genetic background in which we can predict a drug effect. And finally, as well as this inter-individual variation, we can also incorporate sex-dependent differences in gene expression so we can make sex-specific risk predictions. And here's an example of that type of analysis showing a population dose response for hydroxychloroquine. There are families of action potentials for baseline and for 3 ascending doses of the drug. And from each action potential, we can measure that APD90 and plot them in these histograms that I've shown here, where I've started to add simple classification of risk category based on thresholds of prolongation. So anything above 400, I've labeled as amber or intermediate risk. And then anything that crosses that red line of 500, I've labeled as high risk. And this means we're now getting a much richer data set than we had when just considering hERG block or even the single action potential, because we can start to think about probability of risk. Here's a summary of that data for both drugs. So on the top, I'm showing prolongation, plus or minus confidence intervals for the full range of doses being proposed in trials, and predictions for males and females are shown. And under that, I've plotted how those normal, intermediate, and high-risk categories change as a function of dose. And in terms of where we thresholded these categories, that JAMA study I mentioned at the start had significant QT effects in 30%, with a dose that would be right around here, about 1,000 nanograms per mil. And so broadly speaking, the model's working in the right range. What this data shows is that on the whole, there's not too much difference between chloroquine and hydroxychloroquine. Chloroquine would be predicted to be marginally more risky over the specific doses being proposed, so slightly more prolongation, slightly more high-risk events, even though the absolute dose you'll notice is lower. And also for both drugs, the risk is predicted to be higher for females than males. And this is consistent with what's observed in patients for both drug-induced arrhythmias and other QT prolonging disorders. So just in the last couple of slides, I want to show you how some of those COVID-associated metabolic changes might alter the predicted risk across the population. So first, acidosis. If you remember, lowering pH reduced the potency of both drugs. And when those measures are put into the model, they can cause quite dramatic effects on the predicted population risk. So here's the data for a high dose of chloroquine, 1,000 nanograms per mil, which in the male population results in about 70% of subjects falling into either into intermediate or a few in high risk categories. And in the context of that shift to pH 7, which might occur with respiratory failure in COVID, the risk specifically related to drug-induced arrhythmias is almost completely eliminated. So while these patients obviously have some very complex issues to manage, the data suggests there's not a need for additional consideration of drug-induced arrhythmias in the context of acidosis. And the same would also be true for fever, where we saw reduced potency at higher temperatures, if you remember. Though obviously both of these effects, the pH effect and the temperature effect, can be drug-specific, so will need to be measured in a compound by compound basis. Finally, here's an example at the other end of the spectrum where a metabolic change would increase the population risk profile. So I'm showing an extreme example here to illustrate the point. So this is the highest dose of hydroxychloroquine I've been seen in any proposed trial in a female population. So you can see there's very significant prolongation predicted with around 40% in the high-risk red zone. And we're also starting to see morphological change in the action potential, suggesting some of these cases are already on the edge of ectopy. And in the context of hypokalemia, so that reduction to 3 millimolar of potassium, there's a strong shift to the population risk with almost all cases now in the red zone, and actually outright prediction of arrhythmia in about 5% of cases shown in black in the risk plot at the bottom here. So for hydroxychloroquine specifically, additional consideration of potential for drug-induced arrhythmias should be given in patients with hypokalemia, and this is over and above the proarrhythmic effect of hypokalemia itself. So just to summarize overall, I've shown you that simulations incorporating ion channel screen data can be used to develop population risk profiles for potential COVID-19 therapies, as well as obviously other drugs outside of the context of COVID. Suggested that the highest doses of chloroquine and hydroxychloroquine being proposed are likely to be problematic in significant proportions of the population, particularly for females. And this emphasizes the need for QT monitoring in those trials. And of course, since many of them are actually focused on safety because of these concerns, that's probably already integral in the studies. We've shown that metabolic changes associated with COVID-19 alter population risk and likely need to be assessed in a drug-specific manner. And finally, to reiterate that point again, that we've got an assay in terms of the ion channel screen, as well as the simulations that can be turned around quickly and can be applied applied to other potential COVID-19 therapies being explored, with just some examples here: remdesivir, lopinavir, and ivermectin, with these antivirals particularly having a close eye on them for QT effects. So with that, I will just say thank you to the people who have done the work in my lab, particularly Cliff Tebay, who, as I said, he's a new research assistant, and he's knocked over all this work in the last 5 or 6 weeks. And very MacArthur, who's the head of the Victor Chang Innovation Center Cell Function and Screening Facility. Lots of members of my lab and Jamie's lab also contributed to the establishment of these workflows. These guys obviously pay the bills, and I want to take the opportunity to, for some self-promotion, and encourage anyone who has research projects that might take advantage from some really advanced equipment in preclinical screening, in IPS culture, in cell function screening, in metabolomics, to get in touch with the Victor Chang Innovation Center and talk about your project and see if we can help you out. And thank you for listening.
Fantastic. Thank you very much, Adam. Some really important work, and it's amazing that you've been able to do that so quickly despite the lockdown and everything. So we have a couple of questions in the Q&A. The first is from Mark Richards. Have you applied plasma samples with known plasma concentrations of drug from humans at steady state dosing with drugs of interest to the SynchroPatch system?
Do I just speak to answer this?
Yep.
Thanks for your question, Mark. And actually, that is exactly a question that Cliff, the research assistant I mentioned, brought to me the other day. The answer is no, we haven't. So all the plasma concentrations there I've done by estimation, looking at other studies, either for cancer or for malaria, where they've given these drugs to patients and measured plasma values, but we haven't taken plasma and put them on the SynchroPatch.
Another question comes in from Vaughan Macefield. So presumably the studies that you've undertaken so far have been on normal, healthy myocytes. Have you conducted the same channel kinetic studies on myocytes from hearts with various pathophysiologies?
So you're asking whether the ion channel descriptions in the models were based on normal myocytes. They are normal models. They're not recorded from myocytes. They're recorded from these channels expressed in heterologous systems, typically the CHO line I showed you. But another consideration, which may be a point getting to in terms of the risk profile for these patients, is whether they have any underlying variants in any arrhythmia genes that might predispose them to more risk. So we could potentially add those variants in either function or expression of ion channel genes into the model and see how those adapt to the risk profiles as well.
Another question here. Curious to know whether your team have looked at zinc in your assay system.
Anastasia, no, we haven't looked at zinc. The only divalent we looked at was magnesium, which also appears to increase the potency of both of these drugs. I guess the question you're asking around zinc is because of this proposed mechanism of hydroxychloroquine being a zinc ionophore that allows it to enter the cell and inhibit viral replication. But no, we haven't specifically looked at the effect of that ion on our assays.
Okay, we're nearly at 3 o'clock, but another question has come in. If you have any burning questions, please submit them now and we'll try and get a few more in. So from Martin Nash, is anything known about the effects of these drugs at the tissue or multicellular scale? For example, on cell-to-cell coupling, fiber or cross-fiber conductivity?
Thanks for that, Martin. There's not a lot of data out there for chloroquine, hydroxychloroquine, but there certainly is for azithromycin, which has some pretty complex multi-pharmacology, including a kind of biphasic effect on the sodium channel. So initially, it blocks it, but later upregulates it. So that change in your sodium conductance obviously transfers to changes in your conduction velocities. But not for hydroxychloroquine and chloroquine. No.
And a final question, is there a one-to-one mapping between dosage concentrations in vitro and in silico assays, and what is present in vivo?
I'm not 100% sure what you're asking there, Vijay, but so as I said, I've taken from studies in the literature, taken what the Cmaxes would be associated with the various dosing regimens being proposed for COVID, and they're the concentrations that we've used in our simulations and covered in our in vitro assays.
Wonderful. Well, thank you very much, Adam. It's 3 o'clock, so I will bring the meeting to a close and thank Louise and Adam very much for sharing their research with us today. Thank you also to everyone who has joined this webinar. I think we had about 150 people on the call at one stage, which was great. And please join us at the same time next week. We have a webinar on cardiac regeneration with Dr. Nicholas Lamb from UT Southwestern and Associate Professor James Chong from the University of Sydney and Westmead. So thank you, and yeah, see you next week. Thank you very much.
And thanks again to you, Kate and Jim, for organising this. Great session.
Yeah. Thanks, Adam.
Same from me. Thank you. Thanks.